Hello, in today's video we will be talking about the evolution of antibiotic resistance and what changes we can make in order to stop it. Let's start with the basics. What are antibiotics? Many people take antibiotics as a prescription from their doctor in order to help them get better after getting sick. Penicillin was the first antibiotic ever discovered and helped people get cured of pneumonia, gonorrhea, and rheumatic fever. Antibiotics are able to fight against bacteria that cause infection by killing them or stopping their growth. Sometimes, a bacteria will become resistant to antibiotics, meaning that the antibiotic will no longer kill or stop the bacteria's growth. Why should we care about bacteria that become resistant to antibiotics? Currently, the CDC estimates that at least 2,049,442 illnesses, as well as 23,000 deaths, have occurred from bacteria that gain antibiotic resistance. This number is shocking and will only continue to increase as more and more bacteria develop antibiotic resistance. So how do bacteria gain a resistance to antibiotics in the first place? All bacteria have something called DNA, which acts like a toolkit for the bacterium. Certain portions of the DNA, called genes, contain blueprints for certain tools that the bacteria can use. The DNA of bacteria change over time in a process called evolution. These changes are also called mutations. Most mutations are bad for the bacteria because it breaks their tools. Sometimes the mutation will be beneficial for the bacteria and will upgrade the tool or create a tool with a new function. Bacteria can become resistant to antibiotics if they gain a tool which helps them fight against antibiotics. Mutations usually happen really slowly. So how have bacteria been able to speed up mutations in order to gain antibiotic resistance? Scientists studying this question have come up with three methods that bacteria use, including antibiotic tolerance, a faster mutation rate, and the ability of bacteria to exchange genes with one another.
The first way bacteria gain resistance is through tolerance. Imagine this bacterium wants to overcome the wall of antibiotics, but it is unable to because it cannot develop resistance. Now, instead of being a wall of antibiotics, there is a staircase where the amount of antibiotics slowly increases. The bacterium develops tolerance to move up the beginning of the staircase. The first couple of stairs gives the bacterium chances to evolve resistance. This bacterium now has antibiotic resistance and can survive high amounts of antibiotics, indicated by reaching to the top of the staircase.
Here we are depicting proper usage of antibiotics. When antibiotics are taken correctly, the bacteria have a difficult time acquiring tolerance and eventually resistance. Imagine a wall. With no stairs to get over the wall, the bacteria will get killed off by the antibiotics. Now, imagine someone is taking antibiotics for an infection that doesn't require them, or taking the wrong quantities, or not consistently taking them. This gives bacteria a chance to establish tolerance and thus a concentration gradient, which allows them to climb the stairs and cause disease.
This video shows two different experiments that each use three areas of different amounts of antibiotics that were observed to see if the bacteria would become resistant. In the first experiment on the left, the three areas had no antibiotics in the first two areas, and then the highest amount of antibiotics in the third area. The bacteria were not able to gain resistance to the highest level area. In the second experiment, the three areas had different amounts of antibiotics, which were no antibiotics, a medium amount, and then the largest amount. The bacteria were able to gain resistance because the medium area allowed them to become tolerant to the antibiotic and gave the bacteria time to gain resistance to the area with the most antibiotics. This shows that there needs to be a medium level of antibiotics before the bacteria can become completely resistant. Different areas with different amounts of antibiotics are common in the human body when medication is given to a patient. So bacteria is able to gain resistance to these medium areas. This is why scientists are trying to figure out a way to stop tolerance from being gained by the bacteria, because this decreases the time the bacteria need to gain resistance. So removing these medium areas will cause the bacteria to not be able to gain resistance.
One example of how bacteria get mutations is through a tool that almost all bacteria have called MFD. Scientists found that MFD plays a role in bacteria's ability to evolve antibiotic resistance and identified other tools from bacteria that MFD needs to work. When they took out these other tools, they found that they were able to stop MFD from working and therefore decrease the bacteria's ability to evolve to resist antibiotics. In one research study, scientists were able to prove how MFD works with the evolution of antibiotic resistance. In part A of this picture, we can see that bacteria with MFD can resist antibiotics, as seen by the high amounts of antibiotic needed to kill the bacteria over a 6-day period noted by the dark black square showing an amount of antibiotic greater than 6,000. However, when MFD is stopped by removing the tools it requires to work, only low amounts of antibiotics are needed to kill the bacteria in a 6-day period, as shown by the light purple square in Part B, noting the amount of antibiotic close to zero. Studies like this help scientists understand different ways that bacteria create mutations that allow them to evolve to resist antibiotics. So in summary, more mutations lead to faster evolution of antibiotic resistance.
Genes moving around. A resistant gene is a gene that is helpful for the cells to survive against antibiotics. A chromosome contains the genetic information of organisms in the form of genes. A plasmid is a packaging delivering tool for the cells to use. This process starts with a cell containing a resistant gene plasmid and a cell without one. After the cells create a bridge to connect the two cells together, the resistant plasmid is then copied onto the new cell. After separation, both cells will have the plasmid present and the resistance for antibiotics. With no resistant gene, the cell will not be able to survive in the high concentration of antibiotics. Even a cell with a resistant gene on the chromosome will not be able to survive in the high concentrations. When a cell gains a plasmid in the resistant gene, then it will be able to survive. The blue line indicates no resistance present, the red line indicates the population with the resistant gene in the chromosome, and the green line indicates the population with the resistant gene in the plasmid. In this graph, we can see that the green line population continues to survive despite the red and blue line population dying off around day 8. In summary, cells can gain a plasmid from other cells. Cells with no resistant gene will not be able to survive in the antibiotics, and cells that have gained the resistant gene will be able to survive in the antibiotics.
In assessing treatment strategies, a study by Perrin et al. Looked into the effectiveness of using two drugs at the same time, known as bitherapy, versus using one drug after another, known as alternating therapy. In alternating therapy, the bacterium is given enough time to overcome both drugs. This is because they are administered one after the other, giving the bacterium enough time to build resistance to one, then the other. Although this method is somewhat effective at hindering resistance, the study by Perrin et al. Showed a far better alternative. In bitherapy treatment, the bacterium must overcome both drugs at the same time. This double barrier requires more work from the bacterium to build resistance to the two drugs, as it must do it simultaneously. As stated, a study by Perrin et al. Compared the impact of alternating two different antibiotics versus using them at the same time. This shows how the growth rate of the study bacterium, P. aeruginosa, is affected over the course of 10 days as it migrates into a new environment containing various antibiotic treatments. The vertical axis displays measurement of growth, while the horizontal displays the number of transfer days. The top arrow points at the effectiveness of the different alternating methods. The gray line represents administration of rifampicin followed by streptomycin. We can see here that this method is only somewhat effective as bacterial growth increases almost at a linear rate, facing minimal hindrance. The middle arrow now points at the second alternating therapy method. The orange line represents administration of streptomycin followed by rifampicin. We immediately see here that there is a significant increase in bacterial growth on day 4, which tells us that this order of drug administration is likely the least effective at hindering the resistance evolution of P. aeruginosa.
The bottom arrow points at the bitherapy method, in which both drugs are administered at the same time. We see that there is a steep initial decline in the bacterial growth due to the bacteria having to overcome two walls of defense, one wall for each of the drugs used. The cost of gaining resistance to both antibiotics is too high initially, which can be shown by the decline in the bacterial growth. However, over a long period of time, P. aeruginosa is able to evolve antibiotic resistance. It just takes longer when using the bitherapy method as opposed to other treatment strategies. One area of focus in the future to decrease antibiotic resistance evolution would be to inhibit the effects of MFD. MFD is an evolvability factor, which means that it promotes mutations, and most bacteria have this tool to increase their rate of mutation. This allows them to have a better chance of gaining antibiotic resistance. Researchers should focus on developing drugs and other techniques to eliminate the function of MFD and other evolvability factors so that bacteria have a lower chance of gaining resistance. Some questions to think about in the future are: what are the specific types of proteins involved in antibiotic resistance? How do antibiotics affect bacteria in different environments? And do all bacteria gain resistance the same way, or are there different ways to obtain it? Something that we can all do as citizens is that when a doctor prescribes us an antibiotic, we should make sure to talk to the doctor and understand why they prescribed it to us. If we really do need the antibiotic, we should follow the doctor's directions and take all of the antibiotic given to us to make sure that it works completely better.
Thank you for listening to our presentation, and I hope you understand the problem of the evolution of antibiotic resistance.